In this case, instruments that are playing homophonically, so they're playing in octaves or unisons, so they're promoting a single kind of melody, a single stream. And then a composer might split that up so the instrumental texture becomes a little more complicated. Now you have the violins doing one thing, the oboe doing another. So you've created two streams in a polyphonic music. So composers really dance between, or use this idea of streaming to create differences in texture, aesthetics in music. So it's not just strictly a speech phenomenon. In fact, it's not even a strictly human phenomenon. So analyzing the auditory scene is important for all species. You need to know what's predator, prey, etc. Right? So I'll just play you. This is a little mouse. This is frog vocalizations. Hear the bullfrog? Right, and he's but you know, his female counterpart would want to analyze the scene, pull out, extract, figure out where in that scene the mate is relative to all the surrounding cacophony of frogs. Does anybody? So here's the sound clip of this scene. Does anybody actually see the frog? Okay, so here's Kermit. So the idea is here is to try to listen to Kermit and filter out all those irrelevant other frogs. Okay. So in terms of breaking this process down, the computation of it, you can there's some basic acoustic features that really underlie sound segregation. These are just three, but there's many more. One obvious one is just location and space. Sounds that occur over here versus here, you can stream those because they're spatially separated. So location is obviously important for segregating sound, and we have a couple different mechanisms in the auditory system that are really good at figuring out where things are based on timing and level differences between the ears. Okay, pitch is another one, right? So pitch is another way to signal that there's multiple signals in the environment. So a good example is the pitch of human speech. Males tend to speak with lower pitch than females. And that difference in pitch tells you that there has to be two entities there. It's very difficult, if not impossible, to produce two pitches with kind of an organic system. So the fact that you have multiple pitches in the environment, let's say there's multiple things there. You might be able to track one or the other. The other one has to do with kind of the frequency characteristics of the signal, or its timbre, right? If we had, say, two male speakers producing the same pitch, you could still distinguish there's two people there based on the timbre and sound quality of their voice. Okay, so these are kind of some of the more salient acoustic features that we can use, sorry, I'm being blown away here, that we can use to figure out what is in the auditory scene and how many perhaps targets are actually there. So it's kind of a feature decomposition approach. So in general, scene analysis is a really computationally ill-posed problem. There's limited sensory data in the environment, or if one could argue maybe an infinite amount of sensory data in the environment. And to figure out what's there is ill-posed. There's multiple solutions to this problem, so there's not a unique answer. And that makes it hard to put into a computer algorithm, for instance. By the way, you can stop me anytime, so don't forget. Okay, so computationally ill-posed problem, but humans do it remarkably well. Psychologically, it's thought that perhaps some of this ambiguity of what's going on at the sensory input is overcome by applying kind of templates or schemas. Right? Your experienced listeners, you've perhaps been to a cocktail party before, you know what to hone into essentially, what features to extract, and you build internalized a priori schemas, right, which help guide your analysis of the scene. Okay, so that's kind of a higher level, more abstract way to think about how ASA is done. An earlier kind of notion is that it's more of a feature-based extrapolation. We saw those different features that you could use, and that perhaps attention is involved here. That selective attention might kind of drive your feature search. So yeah, you have all these cues available, but you sort of lock on or some become more salient or enhanced because you're selectively attending to those features that promote or give you the most information about what's there. So selective attention itself could kind of limit the scope of search and take a very difficult problem with kind of infinite solutions and arrive at a solution. So there's two basic kind of, I would say, modes of thinking about ASA. One is kind of a primitive approach. One is the schema-based approach. So the primitive kind of school of thinking suggests that we're kind of extracting these acoustic features, maybe putting them together in some meaningful way. That's how we arrive at figuring out what's there. So that's kind of a bottom-up approach. And it's always bottom-up, top-down, right? That's all dichotomies in science. So primitive is kind of the bottom-up analysis. Schema-based is kind of top-down. We're using these a priori, your knowledge, your world knowledge, your templates that you built over a lifetime of listening to guide the analysis. Okay. So one question that comes up is, what's the role of attention in all of this? If there's a top-down, bottom-up interplay, and likely they're both operating, how does attention kind of mix in? Is it a factor at all? Okay. So we're interested in the neural biology of all of this. So we're interested in neural substrates. What's the brain doing to help us figure out what's in the scene? So particularly looking at neural correlates. What change in some brain measure correlates or predicts the behavior of hearing, say, two streams versus one. So not only kind of the physiology, but also particular brain areas that might be involved in this process. If there's schemas, where are they represented? Okay. And a more difficult but very interesting question is, what's the developmental trajectory of this, of scene analysis? What's the uniqueness in terms of species? But what's the plasticity of auditory scene analysis? Can it be changed with experience, or is it kind of hardwired from day one? Okay, so I already showed you that it's not strictly human specific, but today if we have time, we'll look at this idea of plasticity, how scene analysis changes with different types of listening experiences that have occurred over your lifetime. Okay, so keep in mind this idea of primitive versus kind of schema-based, bottom-up, top-down stuff. And when we talk about dynamics of anything in the brain, it has to be biologically constrained. So this is just a kind of a snapshot, a slice through the brain, and kind of showing you the progression, the time course, the dynamics of signal processing as this sound enters the ear and gets all the way up to some perceptual abstract representation. So generally what we find is that as you go lower in the system towards the ear, towards the cochlea, this is a little sensory processor. It's like a microphone. And those representations at a very low level of neural processing tend to be isomorphic with the stimulus, meaning the neural response mirrors the acoustic properties of the stimulus. In fact, you probably have heard some of these brainstem responses I played in this seminar where you play back the brain signal, you can actually hear what was in the acoustics. And we've actually done some interesting experiments where we played people brainstem responses and had categorized the original speech sounds. So you can actually hear almost auditorily what's going on at very low levels. And that's because you have basically a one-for-one mapping between neural representation and signal. Okay, so this is really early stuff. And it's thought to be relatively hardwired experience. Although we've got some data that challenges that. That's maybe for another time. Okay, so as we funnel up, so this, by the way, I'm not showing where the ear is. It's here. So we've gone through the cochlea, we're going up through the auditory brainstem to the cortex. If you get a little higher, you begin to see that the neural representations in the thalamus and early primary auditory cortex are not isomorphic. They're a little bit more abstract. They're not just a reflection of the stimulus anymore. There's something added to them endogenously. And here's where we start to see the first glimpse of experience-dependent plasticity, that the brain can be changed in, this is still pretty early, right, 100 and 150 milliseconds after that input each year. So we do see that language experience, musical training, different types of listening experience can change these kind of representations. They're not necessarily hardwired. And of course, we get to auditory cortex and beyond, where we're perceiving and there's cognition for the signals. And these are perceptual representations. They're very abstract, Certainly highly valuable to experience learning, development, etc. But much more abstract than what you had much earlier in the system. So the challenge of understanding the dynamics of ASA is this. You have an isomorphic representation early, and it becomes more abstract as you ascend. So it's a little bit like dodging a bullet, but it's constantly changing on you as the signal propagates from cochlea to percept. So it can be challenging. But one thing I want you to note is that the dynamics of this, there's always a coupling between space and time. So the signal, we can't talk about the brain solely in space or solely in time. They're highly coupled. The signals come into the ear, and spatially they propagate up, and there's signal processing, layers of signal processing that happens as we get up to cortex. And that happens over time. Right? So space and time are highly coupled when we talk about the neurobiology of anything. So this is kind of the basic framework we're interested in when looking at auditory scene analysis in a couple of experiments, tracing signal representations up the pathway. And the real questions are, when does a percept become a percept? When do we figure out that there's two auditory objects there? Okay, when do we go from these isomorphic representations to abstract categories? Okay, so that's kind of what we're looking at in a couple different experiments, which students read in the paper. Okay, coupling between space and time. It's kind of like a physics lecture, right? Space and time continuum. All right, cool. Yeah, that's your lecture, Booter. Okay, so here's the basic idea. We want to understand neurobiology. We can't go digging around in the human brain. Turns out that IRB has issues with that. So we have to use non-invasive techniques, right? In this particular case, the technique we use in my lab is EEG. So we have electrodes all around the scalp. You're doing something, listening to something, doing an auditory task, and we're picking up at the scalp electrical activity that's kind of propagated to the scalp. The real challenge is figuring out how changes in that electrical activity are producing, predicting, reflecting of behavior. Might be great if you had lightning bolts coming out of your head. Those are big signals. So what we measure is in the nanovolt to microvolt, so really tiny stuff. Okay, so that's the basic way to look at the neurobiology of some perception cognition phenomena. Okay. So here's a little kind of history lesson, getting back to scene analysis and this idea of selective attention, that it may drive which features you're pulling out of the scene. This was an early experiment by Hilliard's group. He was kind of a pioneer in auditory neuroscience and ERPs, but this was a very elegant experiment looking at the effects of selective attention on scene analysis. So here's the task here. It's a dichotic listening task. So you have streams of tones going to one ear and streams of tones going to the other ear. Each one would sound like Morse code, almost, these little beeps, happening very rapidly. Every once in a while, what happens is you'll have you have a set of standard tone pips here and not a little deviant. Maybe it's a higher frequency or it's louder. The idea here is you're trying to have the person respond or detect those oddballs. And the task is, there's two conditions, essentially. In one case, you always have the same input. You always hear these tone streams, okay? But in one case, you're asked to selectively attend to the right ear. The other case, you're asked to selectively attend to the left ear, okay? So stimulus is entirely the same. It's just where you're directing attention, to which stream. So here's any questions with that?
Are the patterns of the streams the same?
It's, if I recall, these were kind of pseudo-random to generate. Yeah. So there's no prediction here. You really do have, it's a very attention, labor-intensive task where you have to monitor one here. Okay. So what happens? Here's some brain waves, yada, yada, yada. You get little blips in the brain. But here's kind of a summary of the results. Okay. So remember, the stimulus is always the same in each ear, each channel. But if I selectively attend to the left ear, tones of the left ear are enhanced, whereas right ear tones, in response to those, are kind of remain the same. And vice versa. When I'm selectively attending to the right ear, right ear tones, when I'm selectively attending to that ear, are enhanced. You get this double dissociation. So although the stimulus is the same, what you have here is a magnification or enhancement of what's relevant, what's being attended to in the stream. And I should say this is, so these tone tips were really short, 50 milliseconds. And you're seeing this effect where they're measuring this in terms of the brain time course, about 100 milliseconds. So this is really early processing. 100 milliseconds after the sound hits your eardrum, you have selective attention kind of enhancing, making relevant signals more salient. Okay. So it was thought before these kind of early experiments that this particular wave here, we call N1, is just reflecting stimulus properties, but what we see is there's actually kind of top-down stuff going on. That attention can actually enhance stimulus coding really early. So here's a pretty simple, but I think pretty nice experiment showing that selective attention can pull out or enhance relevant features of the stream. In this case, it was a frequency manipulation. So attention does play some role in analyzing the scene. Any questions? Any questions there? If you attend to something, turns out the brain is actually enhancing it. So that's kind of important.
Can I ask a question?
Yeah.
Mindfulness exercises that people are talking about today, where you want to listen.
The rushing water.
Yes.
Yeah.
Does that go to that middle phase? Back there, that middle phase, is that what gets affected? You're going down that level? Or
So you're
usually not the icing perfect.
right. That's an interesting. Yeah, I haven't thought about. So you're talking about kind of this background, not a masker.
Pay attention to the sound.
Yep.
Pay attention to your leg. And you really to try to concentrate and total awareness of it all.
Yes.
So
yes.
Is that?
That would for me that would be in the green zone.
It would be there as opposed to
the low. Okay. Yeah. We're I mean that's getting into I tend to operate pretty low in the system actually think about it, but this is more abstract constructs, categories, stuff where you have that might play, it might modulate your perception where you have this concurrent, I don't know, water sound. Yeah. But this is in this kind of area here. Yeah. But that's, no one's looked at it from that angle, I guess. It's a lot of these kind of experiments, we can't get to the blue or the red in humans, you have to use an animal model. You could make cats or chinchillas listen to water rushing, but I'm not sure you'd get the desired results. That's a good question.
Can I just ask a question about the representations?
Sure.
So it looks as though, for you, the representations are the signals, right? Those are the same.
Yeah, we're talking about signal representations, right?
Okay.
Right. How is the brain encoding the acoustic signal? How is it represented?
But when you get up to perceptual representations, are you still dealing just with signals, or no? So what's the representation there?
Good question. So that's the trick. That's difficulty. Is that these tend to look like the signal, and we can make sense of them much easier. But as you get upstairs, you're talking about perceptual representations and such, cognitive representations. You're talking about a very complex, very abstract signal. So it's not a one-for-one mapping. And that is the, you just identify the goal of all cognitive neuroscience, is try to figure out what that representation is that is reflecting perception. It's in the lab, we tend to study neural representations by parametrically varying something, because that's what we can manipulate in the lab. We might look at how this brain wave correlates with a change in some signal property. So it's usually signal driven, or vary multiple parts of the signal, multiple features of the signal, and looking at changes. But as an easy way out, if you want to think of perceptual representations as the abstraction or combination of multidimensional features, then you can start to relate it back to the stimulus properties. But it becomes very difficult at a high level, certainly. But that's a fundamental That's the missing, like, that's the black box, I think, of all neuroscience, is trying to figure out that green box. And really with limited tools. I mean, at school we can measure electrical waves in the brain, at school we can look at it with fMRI, but they're limited. They're really limited. Okay. Attention, important in analyzing the scene. So what are we attending to? Well, that's a good question too. So one notion is this feature-based account, right? We have pitch, location, timbre of the signal, these different acoustic features, and perhaps we are attending or allocating our attention to one or more of those different features, okay, to describe that particular information channel. So pitch, location, all these different features. That's kind of a bottom-up feature-based account. Another one is more of a schema kind of account, is that we're attending to the objectness of the signal. Okay, that attention is allocated to a sound object, a collection of features, or a more abstract representation, rather than just pitch, timbre, timing, acoustics. And so then, this idea of object comes from the visual literature. Right? It's the idea that you might have some objects, right? And as I rotate this, the visual cues are changing, the sensory cues are changing, those features are constantly changing at your retina, but you recognize it as the same thing. So you recognize it as a single entity or object, single category. Those are two competing theories, kind of a feature-based account or an object-based account. And one of the problems with early studies on scene analysis and auditory attention is that they were confounded. So you had changes in the objects, along with changes in the acoustic features. So you don't know which one it is, because they're conflated. So I might have an example would be if I'm trying to segregate an oboe from a saxophone. Those are two different objects, but they also vary in their timbre, in their pitch, in their loudness. Okay, so that's the problem with some of the early auditory attention stuff, is you have kind of a and so this is where we tend to break away from the objectness and look at more feature-based accounts, because we can rigorously manipulate those independently of objectness. Okay. So yes, attention plays a role, but there's some interesting, that's not the whole story, it never is, right? So we tend to orient the sound object through streams, and those are the basic units of the scene analysis problem. But it turns out that attention may operate subconsciously, covertly. So here's, let me just walk this little experiment here. Here's an experiment where the task was, you're listening, there's 3 streams of tones, 1, 2, and 3. And what you're asked to do is attend to this series of 1 tones. There's a little deviant that happens every once in a while. That's the behavioral response. You detect the deviant tone in that stream. So if you take the case first, where there's a stream of tones you're trying to monitor, but an adjacent nearby one, that interferes with your detection and monitoring of the first stream. And what you tend to see is that reaction times are really slow for this clustered hard condition. It's more difficult to monitor the first series of tones. If you space it a bit, so you separate these 2 streams, there's kind of 3 independent melodies, reaction times improve. Right? So there's less competition now. But the interesting condition is probably this clustered easy condition. So you have monitoring the first stream of tones here, but trying to do the auxiliary, you have 2 streams that are clustered to one another. This is your target stream, these are kind of masters, but they are kind of clustered with one. And you actually see a facilitation of reaction times. Actually, you're better in this condition. The idea here is that the brain is automatically, subconsciously route these signals together as the interferers and block them out. And that enables you to monitor your targets much better with some facilitation. So attention here wasn't directly manipulated for the different streams, but you can see that somehow the brain is grouping things whether or not you like it or not. It just does somewhat automatically. So even when there's objects outside of the attentional focus, as in this case, you have some grouping or some clustering of things doing.
So this is kind of the way of thinking about it. We brought attention in at the beginning is more of
Yeah.
the probabilistic recalibration of cues,
Yeah.
which would happen on, like in the speech perception literature, that happens very automatically when you're in noise conditions.
That's right.
So
It's an automatic filtering kind of process.
yeah, which doesn't really require attention most of the time, right?
In fact, and we'll see, it doesn't at all.
Okay.
It doesn't at all. This notion of grouping signals can be done entirely pre-attentively, to some extent. Attention is never out of the game, right? It kind of overrides everything. But you have a certain ability, an automatic ability to group things even when they're outside of the scope, the direct scope of attention. So this kind of experiment suggests that maybe we're doing this streaming somewhat automatically when there's not strictly focused attention per se, that it can happen automatically. So the question that emerges is does perceptual organization streaming take place if you're not actively attentive? If there's no attention explicitly passed. Okay. So it's difficult to ask, attention tasks are kind of difficult, and behavior is difficult to ask where you're attending and where you're not to subjects, because you always have to produce a behavioral response. So one way to look at this pre-attentive kind of segregation stuff is to use evoked attentions. And one way to do that is with a response called a mismatch negativity. And the paradigm is something like you saw earlier. You're presented with a series of kind of standard tones, and every once in a while you have a deviant tone that differs on some dimension, loudness, frequency, whatever it is. So the idea here is that you get a brain response to the standard. That kind of creates a memory trace, highly probable signal. Beep, 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 beep. There's your signal to that. And then every once in a while you hear boop. Right? So beep, 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 boop, beep, 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 boop, beep, 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 right? So every time you hear a standard, that creates some neural response, some neural signature. And the deviant creates a slightly different one. And you can see that in this particular latency range at around 150 milliseconds. You can take the difference between these two waves. You can isolate the difference in neural activity between the standard and deviant. This is called the mismatch negativity, this little negativity that occurs. So the brain, we interpret this MMN to be kind of the brain's automatic detection, deviance detection, distinguishing standard from oddball. Now this grows depending on how different the stimuli are physically. It correlates with perceptual discrimination. Okay, so it's a good, fairly good indicator of your ability to differentiate, in this case, two streams, two tone features. The nice thing about this particular evoked potential is that you can have people fall asleep and still get this response. They don't have to be doing a task. They can be watching a video. So it's kind of the pre-attentive discrimination. Okay, so here's kind of an experiment, early experiment, looking at using this MMN in a stream segregation kind of experiment. So I'll walk you through kind of these are the stimuli here. In one case, you have a series of tones. You're creating two streams here. So you have a series of high tones and a series of low tones. But they're slow paced, okay? So each tone occurs far from in time from its neighboring tone. You kind of get this beep, boop, beep, boop, okay? So that kind of stimulus is really perceived very discreetly or stair-step. You hear a high tone, you hear a low tone, you hear a high tone. So we would not say in that case that you're hearing two streams. You're not hearing the upper melody and the lower melody. You're just hearing serial tones. If you present those tones at a fast pace, so that they occur back to back from one another, you can actually create the perception of two streams or two melodies. I don't have any audio of this, but you can imagine hearing an upper melody like a flute in the bass. You can hear there's two things going on. So the manipulation in this experiment was SOA, stimulus onset asynchronous, just the timing between the tones, a slow pace and a fast pace. And the idea here was that if the brain is doing some automatic perceptual organization, if it's streaming, if I deviate these tones slightly, I should get one of these mismatched responses. I should elicit a response because I've created two streams. The deviance from that should be elicited in this. We wouldn't expect one here because we're not streamed. There's not two melodies. We can deviate a tone here or there. It doesn't change the percept. It doesn't change the two streams because there's just, in this case, one. So the rationale is that if you manipulate or change some aspect of one of the melodies, that was, and you saw in MMN that the brain had perceptually organized those series of tones together. So here's the, yeah.
What if it were a 50 millisecond stimulus onset asynchronous? More in that
Yeah.
box.
Yeah.
Is it still sound like it's 2 yellow?
Yeah, so there's a continuum here of all of this, and I didn't bring.
And the other one
That's-
was 250 millisecond.
Yes, yeah, yeah. So it turns out there is some boundary. You can map these boundaries of when you hear one or two streams based on two things. One is the timing between tones. Tones that are close together tend to be heard together, and tones that are farther in distance and pitch also tend to be segregated. You tend to hear that as two streams. If they're close together, you tend to group them together. These are kind of grouping mechanisms. But in this experiment, it was just fast and slow. There's no intermediate manipulation, but that would be interesting to figure out where the boundary can be found.
Just another wrinkle that you might add to our scenario is that tones were at some harmonic interval and close enough to probably be perceived as chords. Right?
Yeah.
They were at a pitch. Oh, that sounds like a
Yep.
huge.
So there you just created another object. You
Yeah.
created a musical interval out of multiple features. So yeah, pretty creative kind of task.
So what if you put a beat behind the really slow one?
Put what?
Put a beat behind it.
A
Just
beat.
like a drum line.
No, it's
Drum line
pretty
kind of.
slow.
It's
Pretty slow.
super slow,
It's pretty slow.
but
So
even if they're,
yeah.
have a drum line behind it.
That's another added element, though. That would be kind of a lattice that you're putting, a beat lattice, right, against the tones that might help promote continuity of the streaming. But these are really slow. If I played these for you, you would just hear boop, boop, boop. They're not.
I don't think it's a good song. but-
Yeah. Well,
No Grammy for that.
no Grammy. Maybe this Grammy.
Grammy, you don't get the research.
That's right. That's right. I'll take the Grammy. Okay. Okay, so the idea here is we should perceptually organize these. We should see an MMN here if we deviate these. And to kind of give you the answer here, it turns out in the slow case where you don't have streams, you just have this discontinuity. You have no mismatch in activity, so the brain hasn't grouped anything, hasn't detected changes in the melodies. But in the fast case, where you've actually segregated these two things in independent channels, if you deviate one of those channels, you get an MMN. So the brain has perceptually organized things and automatically. So they're really pretty early responses at 100 milliseconds. So this was kind of a neat experiment because it challenged some of the early attentional stuff that perhaps we can perceptually organize or stream somewhat pre-attentive, somewhat unconscious. Oh man, we're terrible on time. Okay, so so a little bit of an intro conclusion here. So we think of primitive or automatic processes and a lot of auditory scene analysis nowadays. And there's other good evidence for this. Infants can stream early in life. There's a bunch of animal studies looking at stream segregation, different neurons along the pathway. You see properties of streaming just beyond code. So it's not clear that attention and high-level top-down processing is necessary to do a lot of the building work in terms of figuring out what's in space. course, the caveat is that attention always is the kind of overrider. In fact, the buildup of streaming, as you start to hear two streams emerge, that builds up over time. In fact, in perceptual studies, attention is a heavy driver of that process. So attention is always at play, but a lot of it's quite automatic. Okay, so that's a bunch of other people's work. That's that's okay. It's kind of a historical overview of stream analysis. So we've looked at this issue a little bit, looking at this hierarchical processing. We're interested in when stream analysis, when perceptions emerge in the brain from this isomorphic perceptual representation. And the way we do this is with a concurrent sound segregation task. Just to speed up, this is one of the things the students read. If you have a tone here, which its harmonics are perfectly integer related, you hear a single pitch. It sounds like this. You hear one tone, one pitch, one object, one string. If you take one of those harmonics and slightly mistune it from the harmonic series, you get something different. It sounds like this. You hear that? You hear a low tone, and you hear this other faint, higher pitched tone. And so if you have people report what they heard, they hear two strings in this case, just by mistuning one of these partials. So you can parametrically, let me skip that a little bit. That's the kind of task we're using to look at concurrent sound segregation. And in my lab, we're interested in this hierarchy of processing. We always do behavioral measures. We can measure cortical activity, brainstem. We've done some modeling stuff with the auditory nerve. We're interested in looking at, as the sound enters the ear, what is the representation of these various hierarchical stages, and when does it become the perceptual goal? Okay, that's the goal. It's much more difficult than I just laid out there. Okay, so back to our stimuli here. So here's our different tones where we have a perfectly single pitch tone, no mistuning. And as we just mistune that single harmonic by different percentages, the behavioral data fall out like this. So what you tend to see is that people always hear perfectly harmonic tone as a single auditory object. And as you mistune, you get a nice categorical psychometric function. At 16% mistune, you start to hear the pitch and something else, so your two objects. Okay, so you can parametrically manipulate kind of sound segregation here. And you heard these two examples just a minute ago. So that's the behavior. You get this nice, this is a continuous change in the stimulus, but you get a categorical change in the response. And there's the reaction times. There's not much going on there, except people are a little bit slower around this midpoint of the continuum, as they always are, because that's where it's a little bit ambiguous. I don't know if it's one, I don't know if it's two. I'm a little slower making that decision. So that's pretty consistent with categorical perception data. All right, that's behavior. And of course, we slap some electrodes on. We record some brain responses. We do some fancy stuff in my lab where we are able to look at cortical responses simultaneously with brainstem. So we're trying to look at kind of this isomorphic and more abstract representation. I won't bore you with all that, but we'll just look at the evoked responses to these mistuned stimuli to see what happens. So if I'm listening to a tuned sound, I get this kind of response, the cortex, and a mistuned response gives us another neural signature. And what we can do is just take the difference between the two to isolate what is different between brain activity and tuned versus untuned, one versus two objects. So consistent with some earlier data, you get this something called the object-related negativity, which is again around 150 milliseconds. And if you plot that guy, the magnitude of this response with this tune looks a lot like the psychometric function. So this ORN, pretty early on, detection of auditory cortex is actually coding pretty well. The degree of this tuning maps pretty well with that psychometric function. So we think that that particular response is kind of figuring out how harmonic a signal is versus inharmonic. If you go later in time, though, so this is more kind of just reflecting this tuning. There's another wave here. You can see the actual raw response. It's a very large wave of about 500 milliseconds. And this later stuff tends to correlate with the salience of the percept, not the mistuning per se, but the salience of the percept. So salient stimuli like the totally tuned and the highly mistuned, where you clearly hear zero, you clearly hear one versus two objects, elicits a very large N5 responses, activity 500. And as the percept gets weaker, in the ambiguous cases and the weak kind of mistuned cases, this particular response. So it's
The last one is comparing two that are mistuned.
you probably can't see them there. So this is the pooling of this of the 0 and 16%
Right.
emissions. This is 2 and 4 in the middle of that psychometric boundary.
Mistuned.
They're mistuned, but they are sometimes classified as 1, sometimes as 2. It's a little bit ambiguous. The point is here, you get early stuff. ORN is correlating with this too. It looks like the psychometric function, but that's also correlated with acoustics. Here, the N5, this is kind of a brain signature that's actually telling us something about the salience of the percept, how confident you are in whether you think that 1 or 2 objects are there. You can start to see dynamics of the brain here. Early stuff is reflecting stimulus acoustics to some degree, and later stuff is starting to reflect the abstract, the salience of the actual percept itself. Okay. All right, flying through. I'll skip all this. We've gone lower in the system, and it turns out that you can predict there's a lot of harmonicity stuff, coding that's happening in the cochlea and the brainstem very early in the pathway. These are these isomorphic representations we talked about. But if you throw everything into a big regression model, as we like to do, we're trying to predict behavior based on a combination of variables, the brainstem stuff we measure, and all of these later cortical responses. What you tend to see is that perception, reaction time, and your classification are best predicted by, you might expect, later cortical activity. Stuff that's happened in 150 milliseconds out. So this is the stuff that's reflecting more decision processes. Early brainstem stuff, while it correlates extremely well with acoustics, it doesn't pop up as a significant portion in predicting behavior.
Could you explain how that regression is run? Because I'm not sure what the parameters would be.
Yeah, so I blurred through that. So what we're trying to predict is a dependent variable is the reaction time or is percent correct, something like that. We've measured something in the brainstem from our ERPs there, the magnitude. We've measured these N5, the amplitudes of the N5 component of the ORN component and their latencies.
So we create a vector that's composed of these 5 parameters.
Yeah, it's a multiple regression. You can see which floats the top in terms of adding a significant contribution to this variance.
And so you literally have the whole time series for each one of those parameters that's being entered as part of this vector.
These are just multiple measurements. So we're getting, from the cortical responses, just picking out what this magnitude is and what this latency is.
Oh, just the magnitude.
Yeah, you could certainly do it for every time point in this series, but that would be.
That's what I was thinking. You might be doing N5 so confused.
Yeah. So the way evoke potentials are usually measured is there has to be some significant deflection from baseline. So yeah, that's a good question. But what boils to the surface is not these more isomorphic, exogenous coding. It's the later stuff that tends to reflect behavior the best, as you might expect. So here's kind of what we've looked at in terms of the different neural structures that are contributing to this mistuned harmonic sound segregation task. So if we move right past the cochlea, right the auditory nerve, what you see is that it tends to code harmonicity really well. So you get stronger responses for harmonic signals and then weaker responses for inharmonic signals. It's kind of a flip of the behavior. So it's capturing acoustics really well. Brainstem responses are also capturing this tuning. But by the time you arrive in cortex, what you see is kind of the flip of the sign. So now bigger is not better. It's flipped, right? So here, early stuff was was declining for mistuned sounds. Declining. By the time it gets to cortex, it likes mistuned sounds. It lights up. Right? So it tends to respond much more vigorously when we have multiple objects in the scene. It's kind of like we've had a transformation here in sign between lower and higher processing. And there's the behavior. So then you start to get into actually looking more like behavioral responses. By the time. So early components of the auditory system are reflecting harmonicity and stimulus acoustics features extremely well. But as you move upstairs, what you tend to see is, and those do not though reflect behavior. It's not until you arrive at auditory cortex and beyond you start to really be able to predict what's going on in someone's actual behavioral response, what they're hearing. Okay. Yeah, so I had some more stuff, but it's getting time. I'm worried if I should get into it or not. But we looked at kind of this hierarchy of processing. When are these features extracted? How are they extracted along the path? What's the dynamics of that? We have this space and time kind of dynamics. We'll talk about cocktail party listening. That's kind of a short time scale. Right? This is just what happens as you're listening to two things and trying to figure out what's there. So that's the dynamics kind of online. But what about longer time scales, kind of meta-dynamics? Can scene analysis, this stuff we're looking at, be changed with different listening experiences, with training, with development? I don't know if I should go there or not. Turns out it can, but, but I think I'll end it there for time, and you're welcome to ask questions about this if you want. But what we're looking at now is kind of uniqueness and plasticity of auditory scene analysis. And it turns out that one of the primary factors in terms of being able to segregate speech from noise is your language background. Okay, so bilingual people have a difficult time hearing non-native speech and noise. And they have perfect hearing. They're entirely fluent in their non-native language, but if you put them in noise, they have a lot of trouble. And so we're looking at that, how language experience can kind of change your ability to filter signals from noise. There's a lot of kind of clinical implications of that, because if you get a bilingual person in the clinic and you're measuring their speech perception, you got to know that they're going to do much poorer than your native speaker. And that would, you don't want to say they have hearing loss, it's just their language background. So language, musical training, both of those human experiences can change the way you filter and extract things from the scene. So I won't go into that. Okay. The end.
All right. 150 milliseconds seems to be a magical time.
Yeah.
And as the higher level objects play a bigger role, that must have some implication on what the minimal kind of psychological time span there is for object recognition.
Yep. Yep.
So it can't be too short. It's got to be like a moment or a duration. It must be around 50 milliseconds.
It can be shorter. It depends on this how complex the signal is. So you can actually identify what a pitch is with just a few cycles of a sinusoid. Okay, and that might be just a few milliseconds. That's a pretty simple example. But in terms of detecting what a piece of music is, or what the instrument was, or who the voice was, there's a certain amount of time, a certain amount of integration you need to do. And I would agree that 50 to 100 milliseconds is a magical number. I think for two reasons. One is that it takes about 50 to 100 milliseconds to start to see these responses in cortex. They have to get upstairs. Yeah. So there's a propagation delay. That's one factor. The other is that you need a certain amount of time to integrate these different features and come up with a solution. And the people measure what's called the temporal integration window. How much stuff do you need to arrive at the solution? And that's 50 to 100 milliseconds in many cases. So there is something not magical about that, but it's just based on sort of the physiology. some
But
ways.
it still keeps coming. I mean,
Yep.
stuff is still.
Also, yeah, it's continuous. That's right.
And so,
So now you're monitoring that over time. That becomes even more complex. Yeah. We just kind of, the laboratory, we tend to think of these, or look at these things in isolation. But in the real cocktail party, you're constantly doing this monitoring over time. So there's not only a dynamic kind of online going on at the moment, but there's dynamic over time. And there's a dynamic, meta-dynamics in terms of, changing this process with different experiences. So that's, 3 layers of dynamics. So, yeah. Question?
Is there any, like, other sensory modalities which help in narrowing down? I mean, it seems like in the cocktail party, part of what helps me to block out other people is I'm standing, seeing the person speaking in front of me.
Absolutely. Yeah, we, if you just are talking about the sound perception, you can study this phenomenon, but one of the major factors in all of auditory perception is vision. And in fact, you normally exploit vision to figure out what a person is saying. And that would be, there's all kinds of studies looking at auditory and visual, the perception of speech with auditory cues, with auditory and visual cues, or just visual alone. You always see a visual enhancement. If you combine the 2 modalities, you do much better than either one alone. That's always the case. It just becomes a harder problem because now you're chasing 2 systems, and one for me is hard enough. But yeah, certainly.
When Deb asked her first question, I was surprised he didn't say something like, at the highest level, the level you kept referring to as upstairs.
Yeah, because
would be auditory objects such as the particular voice that you hear. This person's voice as opposed to that person's voice. Or
Yep.
the particular word that was said. Or
Yep.
the string, the fragment of a melody that was heard.
Yep.
And all of those are abstractions, aren't they? I mean,
absolutely. So
why didn't you say that? I'm just curious.
well, the question is where that so I'm a bit biased because I do my doc work in the brainstem, and so everything above the brainstem is upstairs to me. But we're looking at where those templates or schemas represented. We're doing a little bit fancier analysis with let's see if I have a picture of it with our we have a cool video of not just looking at the signal doesn't stop at auditory cortex. Where's the template stored, especially particularly for speech? So if you look at we can actually do some source localization stuff where we're looking at different patches of cortex like that, just like fMRI with EEG with really good temporal precision. But here is your brain lighting up in response to speech. The first half second, first at real time, and then 4 times slower. So notice all the stuff that's going on after that, after you hear. There's a lot of stimulus encoding. You see auditory cortex blip. There it is, but then there's a lot going on very early in the chain. So your brain is heavily parallel processor. It gets in and it starts spreading rapidly. And in fact, if you look at scene analysis and speech perception, if you compare clean perception, neural activity for clean speech versus noise-created speech, you tend to see a lot of auditory cortex light up, as you'd expect. Broca's area, right? Language cortex is heavily involved on actually both sides of the brain. If you look at where noise dominates clean speech, linguistic left hemisphere kind of goes away, right hemisphere tends to take over. So it's kind of in a noisy speech signal, it's less linguistic per se, perhaps, and so you need compensatory processing on the other hemisphere to kind of help you out. And what I didn't show here is this changes with language experience. So the recruitment of language versus auditory areas is heavily dependent on whether you're a mono or bilingual person. But I'll say that. So, I don't know if I answered your question, I just talked. So, yeah, okay.
So with experience, you're talking about language experience. What are the age effects?
Yeah.
Compared in the study in neuropsych, you get 10 subjects all the same age.
Yeah, that's a good question. We haven't looked at age. We haven't looked at development. We've looked at aging. So the ability to segregate streams in older adults is usually poor. But in terms of the other age end of the spectrum, the young ages of the spectrum, my intuition from some animal studies is that this ability happens very quickly. Within about the first 2 years of life, you're able to do all this stuff. But there is a slight developmental window there. Early in life, and declines later in life. The question is, can you enhance it or change it in the middle of life? And yes, you can with different experiences. All right. Hey, yep, yeah, sure.
One quick question.
Yeah.
Might be a comment, I'm not sure yet. There's this notion that the job of sensation is to kind of extract features out of the environment, right? So the really low-level visual neuroscience stuff is like, this is your orientation detector,
Right.
and this is, then it all comes together and you see a square.
It's gradients and patches.
Recognize an object. And that kind of gets it, tends to be homuncular. So the job of perception is to, and sensation is to break everything down and then reconstruct it in the mind, and then who's looking at it, right? So your work looks different, a little bit different than that though. And I want to hear what you think about this. But it sounds to me like those really early auditory things aren't doing feature extraction necessarily, but they're kind of doing the best they can at preserving the sound as it is and turning it into neural signals. And then it gets kind of mushed around.
Up.
Yeah.
Yeah, I would totally agree with that. But you can think of the, that one-to-one mapping that happens early is still, you can argue, is just a set of features that are being coded. Yeah, I mean,
Getting richer.
Getting richer, yeah, more abstract, more sparse, I would suppose. You're taking the salient, you're taking everything that comes in the system, and that's captured really well, low level, and then you're trying to pick apart the most salient pieces. Because you can't do it all. You can't do it all. No sensory system can. So you have to, in order to cope with the world around us, we have to pull categories out. And one way that's thought that we do that process is to feature extract. So you have a more abstract technique. Like that. So I don't have an answer to that, but I do think the auditory system, in the auditory world, we tend to model off the vision people and tend to talk about feature extraction because that's usually what we can measure in, say, single neurons. We can measure their response characteristics, how they're tuned to frequency or tuned to whatever. It becomes very difficult to put it all together, though. Right? How do you go from a single neuron to then populations and cortex and lobes? So keep us busy for a while.
I just wanted to comment on that, which you just said. That's the limitation of EEG when you measure auditory, the processing of auditory stimulus from EEG. So even after the segregation of the stimulus, what happens at the lower level, but at the end, what you measure a collective response. So it's an integrated response, which might be the pieces of that whole curve that you see, that response might be coming from different areas of the brain, different pockets of neurons. Some responding to high frequencies, some responding to low frequencies, some responding to fast temporal processing, and some to slow. But we are not looking at like if you look at individual neurons, probably you will get that segregated information from separate areas. But ultimately, like you see from the top, from the scan, if EEG is a tool, then you get an integrated collective response, some response.
Yeah. So that's a limitation with the technique, the resolution, the granularity of what you're looking at. But I think your question's more basic. Even if we go down to like the single neuron level, is it just pulling out individual features and is some other neuron integrating, or is that object, for instance, represented in a sparse way? We don't know. The feature count seems to be pretty good because that's what we can study in the lab. But,
Then we have these top-down effects.
and you got top-down, gestalts, right? Gestalt theory versus.
I want to take Grace's question way outside.
Okay.
I was trying to think of least unfair question I could throw at you.
Yeah, give it to me.
But I will play with it a little bit. And in the dynamic systems mode, because this is the way I think about sound and hearing, and the way I teach it in food science. I believe that sound came before hearing. When tree falls in the forest, nobody hears it, there's still a sound. Because sound is physical vibrations in the medium. And acousticians think that way. So I wonder, you know, when you ask, is there something special about 150 milliseconds, and without, you know, without denying that there are certainly neural timelines that would explain that, my question is, could those neural timelines be timed that way because that's such special time in the physical universe?
Yes.
So,
That's a good answer. Well, what is that?
Yes. So I mean, so what you're getting at is rather the brain is kind of in training to its environment.
Yeah, it's adapted to its environment, one likes to think. And so what is it about, it's possible that there's something, I mean, you know, there's this 20 millisecond concept in DOT.
Right.
And it does seem that that specific synchrony or asynchrony between clicks is adaptive in the sense of us locating, doing localization. And I think some of the most sophisticated explanations of speech perception end up getting rooted in simple survival in an acoustic world.
Yeah, that's kind of stuff we're looking at. I mean, if you talk about scene analysis, it can be a very complex issue, cocktail parties and all that. Really what it boils, yeah, speech or not, really what it boils down to is, and you know, so we're kind of looking at it as just this, how system is tracking properties of the stimulus. And so, and when that kind of occurs.
So,
that's an interesting
can I ask
question.
somewhat of a follow-up question? Suppose you have a signal,
Yeah.
you know, and you delayed it. If you started a signal and then an exact same signal 20 milliseconds later.
Right.
What does that sound like?
Really, beep, beep, really quickly, much faster than that.
Not beats.
Oh, okay.
But, like somebody, it can be music.
Yeah.
Or it can be somebody saying a word, but you started out that vocalization or that music, and then you have a delay.
Right.
That same stimulus.
I see.
20 seconds later.
Yeah.
Where you'd expect some sort of share of the perceptual.
Yep.
Could you, what does that sound like?
People have done that with localization tasks. That's called the precedence effect. In fact, if you take a signal and slightly delay it by 20 to 40 milliseconds, you can create, depending on what that window is, how long that time difference is, you can create the perception of an echo, perception that sounds like kind of a weird repetition. If it's within, I think it's 40 milliseconds, you hear it as kind of one conglomerate sound.
That's what I was wondering.
But a funky,
conglomerates.
yeah, you hear it as a single source, but it's funky. There's what's acoustically, there's some comb filtering, which changes the timbre of the sound. So actually, this is an important property in speaker design and trying to get timing and signals in space. Is what is the maximum permissible delay you can delay a signal going to a speaker without it sounding distorted or echoey or some of that. So people have looked at this. I don't follow the localization literature that well, but that's kind of the phenomena you're talking about. You can, if signals are so close to one another, you actually get. I'd have to play it for you. It's a strange filtered, comb filter kind of sound. It changes the timbre, but you may not hear two repetitions.
If the two signals are presented separately on two speakers apart, right, and then it will appear as a moving sound.
Yeah, so there's issues over space too, whether, yeah.
My question is more specifically about the cocktail party. I mean, everybody has experienced it that if you're not talking yourself and you're kind of getting one book of conversation that you're in, you start listening.
Yeah, to the noise.
And sometimes you can keep track of both.
Yes, yes, right.
Maybe you're right. Noises and still.
That's right. Yeah.
What the other person is saying. So I mean, how does that work? That you can keep track of two.
Very complicated. People look at that. So you have these concurrent streams going on. The noise, you should be ignoring, but you're not, and the target. And that that's just a mixture of all these acoustic features. But what the explanation there that people are thinking is that selective attention draws you to either one. So it would enhance if I'm selectively attending now to the noise rather than target, I'm enhancing signal properties from these guys and inhibiting properties from the target. And so you can just interplay. You can dodge attention back and forth and selectively enhance features trying to listen in for. That's kind of like that first experiment, that Hilliard experiment with the dichotic listening. You enhance streams depending on what you care to listen to. But it's an extremely complex process with all the features going on. And yeah, I don't want to think about it. Yeah, that's good though. Thank you.